Hello friends, welcome to my YouTube channel. In this video, we will discuss Punnett square method. This method was developed by Reginald C. Punnett in 1917. This method is basically a diagram. So this is a diagram that is used to predict the genotype of crows in breeding experiments. So this is a diagram that is used to predict the genotype of a crows during or in breeding experiment. In this diagram, first a large square is drawn. Then this square is divided into blocks by horizontal and longitudinal line in between that cross. So suppose here I have to cross between capital T and small t. So if I have to cross between capital T and small t, then this capital T will form gamete, will form capital T and also capital T. So I will write gamete of one parent on the top of this square. Here I am writing capital T above one block and another gamete, that is capital T, above another block. And this parent will form gamete that are small t and small t. So these gamete, I will write these gamete on the left side of square. Small t on the left side of this block and small t on the left side of this block. Now I will write the genotype of a cross or this cross in that block. And what will be genotype? I will take one gamete from this upper side of that block and one from this left side of that block. So here will be capital T from this side and small t from this side. And in that block I will write capital T from this side and small t from this side. In that block I will write capital T from upper side and small t from this side, similar to this. So now I have genotype of cross. So this is a cross between capital T and small t and small t. Now I have a genotype of cross. So this is capital T small t, capital T small t, capital T small t. So this type of diagram, which is used to predict the genotype of crows in breeding experiment, this diagram is called Punnett square. So this is a diagram to predict the genotype of a crows in a breeding experiment. So here we have here we are doing a breeding experiment and we have two parents, capital T, small t, and we will write gamete of one parent on the top of that square and against each block, one gamete against each block. So the gamete of another parent we will write on the left side, similar to that against each block. Now we will take one gamete from this side, one gamete from this side, and there will be our genotype of progeny. One from this side, one from this side, and in that block we also have genotype of progeny and similar to this. Now we have genotype of progeny of cross between capital T and small t. So this is a diagram that is used to predict the genotype of crows in breeding experiment. So this is Punnett square method.